Okay, so that's nice. And okay, so we have our next seminar. So this would be actually interesting because our speaker is from Poland. So basically, Professor Jan Brezovsky. So he's graduated from the Masaryk University in Brno, Czech Republic. And currently he is having a joint position at the Adam Mickiewicz University in Poznan and also the International Institute of Molecular and Cell Biology in Warsaw. So his main interest, I mean, it's quite broad at this moment, but basically it spans from molecular biophysics, computer simulations, proteins, and ligand-transport, which is actually on the main title. So please, now the floor is yours.
Now it's mine. Okay, thank you, Adolfo, very much for introduction. I'm very happy to have this opportunity to present the work from my group to all of you. And as you can see, this very long title, it's going to be about mainly the ligand transport processes and the implication of these processes for protein function. To give it a bit more poetic flavor, I always say that this is basically to take you onto the journey to the enzyme core and back. And here I would like to highlight that depending on your experience, you may not have encountered that many enzymes that have the active site buried deep inside the protein core, but actually about 50–60% of global enzymes has their active site deeply buried. It is relevant to all type of EC classes, all structural scope classes. So you will see that in many enzymes, actually, the active site is buried on the purpose because it provides a very distinct environment to perform their catalytic function. And then if you have the buried active site, what you need for a small molecules, for substrates and products to reach and leave the active site, you need molecular tunnels that allows this transport, right? So in many enzymes, you will see molecular tunnels. In fact, there are frequently quite complex networks of these tunnels that lead to the active site, with distinct tunnels frequently being used for particle function, like one for the substrate, another for product release, maybe water access, and so on. But if you start checking the structure of these proteins, these enzymes in the PDB database, what you will frequently see is a situation where the enzyme active site is kind of separated from the surface because the tunnels are transient, equipped with multiple gated open and closes only rarely. And given that this is a rare event, then it's relatively hard to study experimentally. In many cases, the tunnels and the transport processes are not really accessible to the direct experiments, which leaves us with molecular dynamic simulations. But because these are rare events, as we said, there are frequently multiple gates located along the tunnel. We need really very extensive sampling in these simulations, very long time, many repetitive simulations, and so on. But with that, we can then understand how the transport process happens if we are able to see these rare events. And to be able to study these tunnels, so of course we can always see whether the small molecules is reaching the active site, where does it go? But if we want to see the tunnels from the perspective of proteins, we need to study these empty spaces throughout the simulated ensemble. And then we need the software tools to do so. Obviously, we will not do that by hands. So the most typical and the state-of-the-art tools are using the Voronoi diagrams, which are kind of skeletons of these empty voids, to analyze the pathways. So here is an example of such a diagram where you have the protein atoms as these disks, and between each disk there is a Voronoi edge, and the thicker the edge is, it means that there is more space to the closest atom available. So in such a diagram, then you can quite easily find all paths leading from some cavity, the active site, to the surface of the protein. So starting from the cavity, we define some starting point in the cavity. We find the shortest and widest tunnels. One, there will be maybe a second, that can be used by small molecules to reach this cavity. And obviously, as we care about the dynamics, we need to do this tunnel identification in each of the structure, in each protein conformation seen in the ensemble. We then merge all the identified tunnels and cluster them to basically define the separate ensembles of these tunnels that occur. We can then analyze the tunnels, understand the kinetics of these tunnels, whether they are kind of permanent, whether they are very temporarily occurring. And obviously, it can also help us to understand what's the priority of these tunnels, likelihood of them being used. So with such a tool, we have been able to study various proteins and see what are the kind of the roles of the tunnels. So obviously, the tunnels allows the ligands to enter and be released from the active site. But it can also help us to select only the cognate ones. So there is some selectivity mechanism that not all ligands can reach the active site. So the selectivity is another function. Then in some cases, when you have these multiple tunnels, as seen on the figure, they can help you basically synchronize the reaction in space and in time, that basically the reactants will come from the proper direction and touch in the proper orientation that is suitable for the reaction. In some multi-enzyme complexes or multifunctional enzymes that basically catalyze multiple reaction steps, what you can see is an arrangement where there are multiple active sites in the structure, and these active sites are connected by internal tunnels that basically helps us to prevent the release of potentially toxic intermediates that occur during this reaction, and also to safeguard the liable intermediates from the hydrolysis outside. So by traversing and being channeled inside the protein structure, the cell is safeguarded, and it's possible to basically perform very complex reactions. And finally, the enzymes tunnels are also responsible for basically control of the solvents access to the cavity. So for many reactions, it's actually beneficial not to have that many water molecules inside the cavity. So the hydrophobic tunnels helps us to get the hydrophobic substrates in, but leave the water molecules outside that can be harmful for the reaction. And when we think about what happens if we introduce mutation to the pathways, so having an active site in yellow, then there is this green tunnel, and there is a so-called bottleneck residue, right? The residue that is forming the most narrow part of this tunnel. And if such a mutation residue is mutated, it may alter the properties of the permanent pathway, modifying the selectivity. Maybe more of the substrate, different compounds can then reach the cavity or less. If you can introduce a gate, so basically changing the permanently accessible pathway to just transiently available one. If we introduce some large bulky residue there, which has a consequences of improved solvents resistance and stability, the ligand will be more longer available for the reaction and so on. But of course, always there is a downside of that it will be harder for the products to leave the substrates to enter them. And the gate can be removed if it exists, turning transient to permanent tunnels. Again, that's an improved transport. And if we modify the gate or the residues that helps to stabilize the gate, we can change the selectivity range and so on. So this is obvious. One can always imagine that these are typical consequences of the mutations, considering the function of these tunnels. This is no surprise. So next, what we did is we follow the famous quote of Richard Feynman: what we cannot create, we don't understand. And we decided to introduce the new functional tunnel into the dehalogenase enzyme. So this is the dehalogenase, typical alpha-beta hydrolase fault, around 300 residues, so quite easy to handle globular enzyme. It has well-characterized reaction mechanism that involves substrate entrance, then there is a nucleophile attack, formation of covalent alkyl enzyme intermediate that is then hydrolyzed, and then the product leaves the active site. Two products, there is bromide ion and there is bromo alcohol in this case. We have also pressed steady state kinetic data, so that allows us to see what are the rates of these individual steps. So we can see the chemical steps, the nucleophile attack and hydrolysis, and rates of the release. We cannot see the substrate entry, which is super fast here. And what you can see or realize is that the product release here is much slower than the chemical steps. So it's limiting the overreaction capabilities of this enzyme, and that makes it really a cool system to study the transport in. And to engineer a new tunnel, we've decided to adopt this particular strategy. First, we will limit the access of this major primary tunnel leading to the active site to help us to provide us with mutant that has a lower background so that we can see the effects of the newly opened tunnel, newly created tunnel. So by limiting the accessibility, we will create a LINB closed system. And in this system, we will study putative potential tunnels that are really of sub-angstrom width, and that might be a good target for engineering to open them. So basically, these are non-existing, non-viable tunnels, some empty spaces that cannot be traversed even by a hydrogen atom, but are good templates to target them and to engineer them towards the open new tunnel. So targeting this particle tunnel, for example, we will perform directed evolution and hopefully reach the fully open tunnel, so introducing a new tunnel. So how it went in practice, to create this negative control, we've analyzed 12 crystal structures of the wild type, and we have been able to find out that there are 3 tunnels that are quite prevalent, P1A and P1B. These are 2 branches of the major tunnel. There is P2 tunnel, which is auxiliary. It's really much longer, much narrower. It's frequently closed. There are multiple gates along its way. And the approach that we used to close or limit the accessibility of these major pathways, we introduced the leucine. We mutated this leucine in here to tryptophan, so introducing bulky residue, but that basically can also form a hydrogen bond with the second helix on the other side, creating a gate in this system. And what we observed is that indeed, by introducing this gate, the accessibility of the active site was markedly decreased. The release rates drop down. At the same time, what we observe is also significant increase in the nucleophile substitution rates, which makes sense because having a hydrophobic gate will also stabilize the ligand in and restrict the water access while the substrate is inside. So this makes sense. And the hydrolytic rates were just a little bit decreased, indicating that waters have some trouble reaching the active site and acting as a co-substrate, right? So having the negative control that has reduced transport rates, we move to the engineering of this new tunnel. So having this system, we have searched for very narrow tunnels that are present there. You can see some lines indicated in various directions that can be a proper choice. We have used a probe radius of 0.7 angstrom. So these are these very narrow tunnels. And we've decided to try to engineer the path in this direction because this is relatively short. So we don't need to introduce many mutations in principle to open it. And it's orthogonal to all the relevant known tunnels. So basically, there is a hope that by introducing mutation in the vicinity of this tunnel, this putative tunnel, we will not affect the primary tunnels. So if you have this look on the tunnel in the detail, so there are 2 bottlenecks of this tunnel in yellow. There are 4 residues that are forming first bottleneck. So tunnel radii along the line, starting from the cavity, going outside. We see that there is this one constriction about 0.7 angstrom, and then there is the second constriction, second bottleneck. Both of them need to be obviously engineered. So to engineer this, we've checked the residues there, obviously, and selected 2 residues forming the first bottleneck for mutagenesis, and the bulky tryptophan from the second bottleneck. Because we don't know how to engineer generally really good tunnel, we've used saturation mutagenesis and directed evolution, simultaneously saturating all three positions using degenerate codons to introduce smaller or original residues. So that there is always a chance to get from 1 to triple site mutants. And upon screening for improved activity with the substrate, we have obtained this most active mutant that contains this tryptophan is mutated to alanine, phenylalanine to leucine, and isoleucine to leucine that we call in B-open. And it has the following experimental characteristics: greatly improved product release, much better actually than the original wild-type protein, which was surprise. And it maintained really good nucleophile attack rates. And curiously, also, it has a significant improvement in the hydrolysis activity on the second stage of the chemical reaction. So this is really basically one of the best enzyme in the whole family with the highest rates, and we've achieved the improvement. And the question is then, is it really because of the tunnel engineering? So we've performed with the group of Professor Kuta Smatanova from České Budějovice, crystallographic analysis, and they were able to obtain high resolution crystal structures for the wolf mutants. And what they see that in this closed mutant, this putative tunnel that we wanted to engineer, this P2, was not affected, and that there is an increase by 0.5 angstrom in this open mutant. So we can see a well-defined tunnel in the crystal structure. And now to understand how these tunnels are utilized, we finally moved to the things that we like, molecular dynamic simulations, and we studied the utilization of these tunnels by ligands. So first, what we performed was standard molecular dynamic simulation without any ligands, and basically just check that this new P3 tunnel is used by water molecules. And then we follow up with accelerated MD simulations with the products where we observed that both bromoalcohol and the bromide ion are actually using this tunnel. So it's indeed a functional tunnel capable of ligand transport. But what was a little bit disappointment is then when we check what are the actual amounts of transport. So what is the probability that the tunnel is being used, for example, in this case, for a product release? What we can see that, yes, indeed, this P3 tunnel acts as an auxiliary conduit for the product, but it's really marginal. It's like an 8% probability that is being used. The originally primary tunnel P1A is still the primary tunnel even in this mutant. So it's not really enhancing much the actual transport property. It's functional tunnel, but still the original tunnel is the major path for ligands to be released, which is curious how it can help then with the release rates. When we check the water transport, we can still see that, yes, P3 is being used for water transport, but again, is a marginal, it's just a conduit for a couple of water molecules, and a different tunnel is primarily used. But what I would like to highlight here, this is the situation when there is no substrate bound. So if you think about the improvement on the product release, what we should actually study is the case where the products are bound in the active site, right? So there's the situation where we have the active site and there are these bound primary positions of the product molecules, and they are actually overlapping and limiting the availability of the P1B tunnel, P1A tunnel, these primary ones that are used in a free system for water molecules. And this is not the case for P3 or P2 tunnels, these auxiliary ones. And actually, when you see how many waters or what's the probability that these products will be solvated by water molecules through individual tunnels before they are leaving. So the solvation by the water molecules is critical point. It helps unbind the products from the catalytic machinery by competing for hydrogen bonds, and then the products can leave. And in this situation, what we have observed is that actually the P3 tunnel and the P1A tunnel are of almost equivalent importance, right, for this pre-release solvation. So it's very important to consider the proper situation in which these water molecules are. So a couple of water molecules on average, but in the right time, coming from the right direction, seems to be very helpful with the release, and are responsible for much better release, but also most likely this site accessibility to the catalytic machinery helps with the observed improvement in the hydrolytic rates. They provide the water molecules throughout the catalytic cycle as needed. So this is really super cool, right? Few water molecules from right direction can have a tremendous impact on the enzymatic function. But if we want to really say that we understand and we can utilize it to prediction of these putative tunnels that matter, both for protein engineering, we may want to engineer other enzymes like that, or we may want to understand the gain of function mutation in the settings of disease etiology. So what are the putative tunnels that might be opened by 1, 2, 3, by a couple of mutations that we have seen here? There are 3 quite conservative mutation with huge impact. One can imagine that if you have putative tunnel in other enzymes, there is a mutation. Improved activity might be actually harmful for the organism. It may also disrupt the activity. So how do we predict these putative tunnels? And then when we were thinking about it, we revisited our simulation data and realized that actually we've detected also some water transport through this putative tunnel, this very narrow tunnel, already in the wild-type simulation. So we are thinking maybe we can use a water transport through these very narrow tunnels as a kind of a marker of their viability. But then the question is, what are the actual requirements for the tunnel to really conduit water molecules? Because on average, we see 0.7 angstrom in the crystal structures. That seems to be very narrow on the radii, right? Water molecule is 1.4 angstrom, 1.7. So that basically brought us to the problems that we need to study really transient putative tunnels, and we need to push our ability to analyze the simulations much further. We need much longer simulation and actually many simulation to be able to thoroughly analyze narrow tunnels and their openings, closing in the large ensembles. So millions of tunnels to be analyzed, hundreds and hundreds of thousands of water molecules to be tracked, and whether they are moving through these tunnels or not. And that basically brings us to some technical challenges that needs to be overcome here. That if we have many tunnels, to be able to cluster them when we analyze them, we have, and that has an enormous memory requirements, and basically disallows such studies, just meaning that we need beyond the state-of-the-art tools. And here we have provided 2 technological improvements a little bit. So first is TransportTools library for high-throughput analysis of MD simulation that basically helps us to deal with the large number of simulations, right? You can perform these days in parallel hundreds or thousands simulations, properly capturing many rare events of the gating or ligand transport, but then you will have very different simulation sets, right? Each simulation will show you different tunnels. Sometimes it's open, sometimes not. This is the real life situation. And we need tools that helps us to basically integrate all this information together and basically integrate the tunnel information, the empty spaces in the proteins with their actual utilization by small molecules, waters, ligands, and so on. So for that, we have developed TransportTools library that allows us to combine various ensembles from different MD simulation, tracing of the ligands together, provided us with their ensemble geometry, critical residues, and the utilization by water molecules. It has also comparative analysis that we can study the systems after mutation, what changes in these tunnels, and so on in a very convenient way. And just to highlight, this comparative mode is actually quite interesting thing is that it's not only for analysis, but can be used also as an enzyme engineering tool. So if you would like to perform ensemble-based design of pockets or tunnels to engineer, let's say, enzyme dynamics itself, we can compare the properties of the pockets or these tunnels in ensembles of evaluated mutants. And as a use case, I would just like to highlight, we have recently done so, engineering a transient binding pocket in penicillin G acylase to different bacterial signaling molecules. And with these tools, we were able to evaluate structural ensembles of around 700 variants, finding six promising designs, and actually 3 of them highlighted here were quite successful, getting really nice activities with different signaling molecules of different pathogenic species. So it can be used also as a design tool. The second thing that we need to consider is, okay, we can deal now with many simulations, we can integrate and see things in the comprehensive way, but we also need to be able to analyze long simulations with very narrow radii, very small radii, to be able to see the narrow putative tunnels and so on. And then the problem is, just for example, if you have 10,000 frames from 100 nanosecond simulation of these dehalogenases, and you would use 0.7 angstrom radii, what we get is around 200,000 tunnels. To analyze those, you need 30 hours runtime and 15 gigabyte RAM. That's not so bad. If you have 30,000 frames, you already need 70 gigabyte RAM, and so on. So it very rapidly becomes unfeasible to really perform the clustering step. So the typical solution would be to use larger probe radii, so seeing fewer tunnels, but that's not helpful for us if we want to understand what tunnels are being used by small molecules, focusing on these narrow ones. The second approach is to use only skipping some frames from the analysis, right? So, for example, we will check only every 10th step, but then it means that there is a good likelihood that actually we miss the open rare event with the open tunnel. Yeah. So from this perspective, we find out that there is another alternative that we call divide and conquer, as is typically used. And basically, it means that instead of analyzing a whole trajectory, we split the trajectory into the slices. Each of them we can rapidly analyze with Caver, so we can control for the size of this trajectory to be really easy to analyze the tunnels there, and we just then use the transport tools to merge all this data together and generate one long consistent simulation. This is really efficient. As you can see, you can have in serial settings as a Caver, we save quite a lot of memory, or we can run the simulation with a little bit higher memory consumption in parallel, but then we can reach really significant speed up. So it really helps us now to basically analyze however long simulation we have without compromising on the probe radii or and seeing every individual frame that we need to see all these rare openings. And just to highlight here, basically the description of the tunnel network using this divide conquer and the Caver analysis, it yields essentially the same insights whatsoever. So there is no compromise in the accuracy, but it's very effective and open us a possibility to actually study such events. So having these tools, we've embarked on the way of finding out what is the minimal requirement for really water transport through these tunnels in the enzymes, basically investigating how water molecules are reaching the cavities. So we also internally like to call it squeezing water to the limits, seeing what are really these narrowest places that water can go. In here, we use as a model systems 3 members of hydrolase superfamily. Hydrolases are very relevant from this perspective as they need the water molecules for this for their reaction as a co-substrate, helps the release of the substrate, and so on. We've studied haloalkane dehalogenase, epoxide hydrolase, and lipase from various bacteria and from potato. And we have used here adaptive sampling simulations. So 5 simulations of 100 nanosecond long are run first. We then analyze them, find what is the least explored conformations. From them, we restart in the next epoch. So altogether, we run 50 simulation in this adaptive guided approach, reaching 5 microsecond sampling for each of this system. Then we've used K-ver with the divide and conquer approach to really see the very narrow tunnels there of at least 0.7 angstrom and larger, but this is relatively small probe, requiring us to analyze over 10 millions of tunnels altogether. So it was relatively huge data set. And we have been able to trace hundreds of thousands of water molecules there and assign them to individual tunnels. So when we have then looked onto the properties of these tunnels that are being used for the transport of the water in these individual systems, we can see that obviously the tunnels are, the water mostly goes through the very widely open tunnels, but curiously, around 20% of the water travels also through tunnels that have at the time of the transport event dimension below 1.4 angstrom radius. So it's essentially narrower than the water molecule itself. Yeah? So in some cases, we see water molecules going, there are few, but also with the tunnel radius of 0.7, 0.8. So these are the situations that basically we are observing, which would basically mean that we have underestimated the number of the tunnels that actually can serve as an access points to the buried active sites from various directions, actually. But it's also a question, so how comes that the water can squeeze really through the tunnel that is smaller than the water molecule itself? And here, what we've realized, analyzing it, is that the narrower the tunnel, the more hydrogen bonds are actually formed between the protein and the water molecules that is passing through this tunnel. We can see that it's quite common to have 2, 3, 4 water molecule hydrogen bonds with the water molecules in these narrow tunnels, unlike no hydrogen bond, or maybe 1 in these very widely open tunnels. Yes, so these multiple hydrogen bonds stabilize water transport and basically allows to compensate for the steric repulsion. And essentially, we believe that the narrow tunnels with a lot of hydrogen bonding capacity represents an interesting identifier of these functionally relevant sites. And more importantly, these hydrogen bonds are formed with the backbone atoms of these residues, which means that this is not a feature of the particle enzyme that we investigated, but generally these are going to be conserved within the protein fold. So whole protein families will have the backbone atoms frequently placed in the similar position, allowing the water transport through these narrow tunnels. Okay, and to give it a more practical view, we have investigated human epoxide hydrolase pathology, where there is a mutation of the glutamic acid at the position 470 to glycine. It's the position that is quite remote from the active site, and it's determined to increase catalytic activity of this enzyme quite significantly, and is associated with higher incidence of stroke. This mutant, when analyzed, basically the ensemble behavior is very equivalent. It's the active site is not perturbed. There is nothing really happening interesting. The only thing that you can detect is the increase in the flexibility of the region quite far from the mutated site. So this is this purple region in the structure there. And when we had a look into more details on what's happening there, we actually find out that this mutation and increased flexibility results on average increase of the 3 quite narrow tunnels. You see they would be originally considered as irrelevant, below 1.4 angstrom radii, cannot be used even by water. And there are relatively small increases in the mutant. but still not really leading to any significant openness of these tunnels. But even this very small change in their dimension results in quite tremendous and exaggerated, I would even say, transport of the water through these three tunnels. In some cases, thousands of water molecules can pass, while in the mutant, in the wild type, we have basically observed no utilization for water molecules there. So this very small increase in the radii of these narrow tunnels results in a rewiring of the water transport in this enzyme active site, altering its viability for the catalytic action, similarly as we have observed in the previous project. So in this way, these tunnels, narrow tunnels that are utilized by water molecules are indeed interesting indicator of the mutation hotspots that can lead to gain of function, mutations, and be responsible for various pathological behavior. So generally, the take-home message from my talk, I would like you to recall that the tunnels are really critical for different function of enzymes, and it's really prevalent among many global enzymes. The mutation can open entirely new tunnels into the enzyme active sites, and these are the mutations that are not really somehow how to say, expected to have a tremendous effects. They are conservative: leucine, isoleucine, phenylalanine to leucine. In many cases, these are very small conservative mutations, and they are placed in the regions that you would generally deem as functionally irrelevant. There is nothing, right? Far from the active site, no access path seen, but there might be a tunnel that is putative and can get open, or there is a transient tunnel that you don't observe at that point, and it can have a really profound functional impact. The water transport can occur via narrow tunnels, and it can be the tunnels that are much narrower than the dimension of the water molecule itself. And with the tools that we have, with the resources that are available these days and advanced molecular dynamic simulations, we can use these computational tools to identify tunnels and the transport processes even in this very regimen, and use it to target the tunnels and engineer the transient or even the putative ones. But there are still some challenges that remains before we can harness the power of the tunnels and the simulation that helps us to understand them completely or more completely. And just three of them that I would like to highlight that are perhaps the most unsettling these days. It's that there is a limited accuracy in the tunnel clustering on the large data. So what I mean is that if you cluster hundreds of thousands of tunnels in a relatively short simulation of moderate proteins, we have already seen that it can be quite demanding. And for that purpose, the current tools are forced to use simplified approximation of the tunnels during the clustering. So when we calculate the distances between each of these individual tunnel, just the 4 points of these tunnels along its path are used, which works quite reasonable for short simulation with well-defined tunnels. But if you move to the regimen of the narrower tunnels and use much longer simulation to see the rare openings, the less discrete tunnel space is generated, right? You will observe the tunnels almost everywhere. So at that point, what you typically observe is that there are incorrect composition of clusters, these should represent a single path that ligands might use. But what you can see if you focus on the tunnel mouths, the exit parts on the surface of the protein, you will be able to see that there are three distinct paths, some of them very different, formed by many different residues, different length, different dimension, that are considered as a single path because of the clustering. And that is, of course, barring the proper interpretation of the biological function of these tunnels and their utilization. So we need indeed some dedicated clustering approaches to develop that will reflect the particle problem in here. The second thing is water turns out to be super interesting probes to understand the ligand transport, and by themselves they are functionally very important for most functions of many of the enzymes. And of course, you probably know that there has been a huge debate about what's the proper water model, what's the proper parameterization, as we use fixed charge models in most cases, and so on. We have seen that these intrinsically disordered proteins are, for example, very sensitive to water models that you use. So we have been wondering that these tunnels are really also very dynamic entities, and they are basically exposed to the surface, to the bulk water and the first solvation layer quite significantly, right? Because they have to lead up to the surface of the protein. So we have a look onto the role of the water models on the behavior of the tunnels in 3 dehalogenase variants. And we use TIP3P, which is one of the most commonly used water model, and OPC, that is one of the very optimized model, 4-point water models that quite nicely approximates the bulk water behavior. And what we have seen that the simulation in both of these systems, with these 2 water models, provided quite equivalent topology of tunnel network. So we have seen the same tunnels and basically the same priority of these tunnels and so on. But what is different there is that in this OPC water model, so orange here, we frequently see that the tunnels tend to remain open for a longer time. So that it's not that it opens more frequently, but when the tunnel is open in OPC water model simulation, then it remains open for much longer period of time, very likely changing the kinetics of the ligand transport. So thinking about this altered kinetics, we have investigated what does it mean directly for water migration through these tunnels. And we have generated 5 groups of simulation with restrained dimension of the primary tunnel that is used in this dehalogenase. So we have basically group 1 simulations that have quite narrow tunnel, group 2 a little bit more open but still below 1.4, group 3 that is more or less around 1.4, 1.5 dimension, so it's already more or less quite suitable for water transport. And then we have 2 quite widely open tunnel dimensions. We restrain the simulation, so we basically are not now checking what happens with the tunnels, but how different water models alter the rates of the water transport through the same type of the tunnels. And what we see, no matter what's the tunnel dimension in here, TIP3P water model overestimates systematically the amount of water molecules, the ease, the transport rates quite significantly. And this is, of course, connected by the fact that the TIP3P is generally has generally higher diffusibility and can reach the active site quicker. And interestingly, in this wider tunnel settings, it also allows more water molecules to traverse the channel simultaneously. So from this perspective, generally, use of OPC water model is preferential, but of course it's a problem. It's a 4-point water model and it makes the simulation quite expensive. So it's quite tricky, but if we care about the rates of the ligand transport, then the higher quality water model is needed here. And just to say, factually, we know that these rates of the TIP3P are overestimated, because there is a similar study performed on the aquaporine where the osmotic pressure of TIP3P is also like 3 times larger than the other higher quality water models. So we need to run more expensive simulation, unfortunately, for a long time with very enhanced sampling to properly study the system. So this is a little bit of a challenge that hopefully the advance in hardware will mainly solve for us. And the last thing is considering that we have all these very demanding and computationally hungry requirements to study the tunnels, how we can use those tools to establish the presence of functional tunnels on the large scale, right? We have 200,000 structures of proteins in PDB database and millions in the AlphaFold models in the UniPro database. So how we can start really annotating properly the transient tunnels on the large scale. So generally we will need to develop some alternatives to rigorous simulation that will allow these high throughput predictions. And most likely it's with the current trend, one would say that use of machine learning and AI models can be useful, but for that we'll need the large data set of tunnels that can be used to train those models to recognize where, for example, in your crystal structure, likely functional tunnels will appear. And of course, then we need MD simulation, high quality MD simulation with proper water models, and none of the groups can really generate such outcomes themselves. And of course, the problem is exaggerated further by problematic sharing in MD simulation data. We have terabytes and terabytes, and how to exchange and share. Here I would like to highlight that there is quite nice initiative, this Miasto dataset, where this is a community effort where they perform quite many QMM calculation on the ligand and studied their complexes with various proteins, 17,000 protein ligand complexes with simulations. This is really cool data set and might be useful for the tunnel analysis, but unfortunately at the moment it's relatively short. So it's useful to see well-defined tunnels, but it's not that helpful for us at this point to see the transient or putative tunnels and really understand where the mutations can have a huge impact or not. And it's using TRIP water molecules. Obviously, it's huge effort, so saving on the simulating of the water makes sense, but it's also hampering the use for the tunnel detection and the training of the models in the end. And more or less, of course, this comes from the databases of the protein ligand complexes that are crystallographically derived. Not all of them have a buried active site in the tunnels. Many of the complexes has also exposed sites and no tunnels. So generally, it looks like that to push further towards high throughput analysis of the tunnels and separate efforts towards compiling the tunnel-centered data set of simulation of suitable systems will be needed, unfortunately, before we can really say routinely here, there are the tunnels of importance, beware or use it for your engineering or as a target for the drugs. Okay, and that brings me to the end. I would like to just thank you, say thank you to my group members that contributed and produced most of the data, to our collaborators from Roche Laboratories that provided experimental data, and obviously all the funding agencies allowing us to have the fund that we have now here. Okay, and that's it. And thank you for listening to my talk.
Thank you, Jan.